Now, in the sequence alignment, let us understand the third one, sequence alignment. In that, the third method we are talking about is dynamic programming for global and local alignment we can use. This technique of dynamic programming can be applied to produce global alignment via Needleman-Wunsch algorithm and local alignment via Smith-Waterman algorithm. In typical uses, protein alignment use a substitution matrix to assign a scores global or local alignment. Global alignment is Needleman-Wunsch. And the local alignment is Smith-Waterman algorithm. Protein alignments use a substitution matrix to assign scores to amino acid matches or mismatches and a gap penalty for matching an amino acid in one sequence to a gap in another. DNA and RNA alignments may be using a scoring matrix, but in practice often simply assign a positive matrix score or a negative mismatch score and negative gap penalty. In a dynamic programming, we will try to take any sequence any sequence if you take A, T, C, G, sequence 1, S2, T, T, C, G. If we take any sequence, if there is a match, if T is matching with T, then we called match. If A is not matching, then we will call mismatch. In dynamic programming, we always take, if it is a match, if any nucleotide is matching with any nucleotide, we will call it as a match, and the match score is always a positive value we will take. When you take a mismatch A and T, if you take A and T, A is here in the sequence 2, sequence 1 comparing with sequence T. When you take this, then it is called mismatch. Then mismatch always we will take as a negative value. It is an assumption. And here you can see there is a gap. If you assume, if you keep the gap here, then the alignment is best alignment. Then if you introduce any gap to the any sequence alignment, the gap score might be -2. Substitution matrix scores to amino acid matches or mismatches and a gap penalty for matching an amino acid in one sequence to a gap in other. DNA RNA alignments may be used as scoring matrix, but in practice often simply assign a positive match score, negative mismatch score and a negative gap penalty. In standard dynamic programming, the score of each amino acid position is independent of identity of its neighbors, and therefore base stacking effects are not taken into account. However, it is possible to account for such effects to modify algorithm. A common extension to standard linear gap cost usage of different gap penalties. This is about dynamic programming. Dynamic programming. Dynamic programming can be used for dynamic programming for sequence alignment can be used in pairwise sequence global alignment and local alignment. Global alignment is end-to-end sequence alignment. This is a stretch of sequence. Compares one whole sequence, global alignment compares with whole sequence with other entire sequence, whereas local alignment computationally, but global alignment is computationally expensive, and local alignment uses a subset of sequence and attempt to align it to subset of other sequences, and computationally less expensive local alignment. Dynamic programming solves complex problems by breaking them into simple sub-problems. Complex problems into sub-problems. It will a whole set of sequences assumes as a complex problems and try to break into small problems. Small set of problems. Problem can be divided into many smaller parts. Needleman-Wunsch was the first proposed this method. Needleman-Wunsch was proposed. Initially, it was applied to global alignment. Later, it was applied to local alignment by Smith-Waterman algorithm. Needleman-Wunsch described a general algorithm for sequence alignment. It is a general algorithm and maximize a score of similarity to give a maximum match. Maximum match, largest number of nucleotide that can be matched with others. That wanted to quantify sequence similarity between two sequences. If you after substituting match, mismatch and gap, you will be getting the whole matrix, and when if the maximum matches are there, maximum score will be there. That is what a dynamic programming Needleman Winch will be explaining us. And when we come to the dynamic programming steps, there are three steps. First, dynamic programming steps, there are three steps. One is substitution matrix calculation. And traceback method and alignment. Dynamic programming is widely used in bioinformatics for the tasks such as sequence alignment, protein folding, RNA structure prediction, and protein DNA binding. And it was developed in 1970 by Charles Delsey and Georgy Guski and Alexander. First dynamic algorithm for protein DNA binding was developed in 1970s by Charles Delsey, Georgy Guski and Jasa Detalvi, USSR. Next, let us see dynamic programming steps. The first step is substitution matrix and initialization. Initialization, in this we will try to start with, there is in the substitution matrix there will be m plus 1 n plus 1 matrix will be there. If the sequence length is 5, then it would be the total matrix size would be the total matrix size, it would be 6 by 5. This is what initialization. Let us see how to see that, how to do that. The first step in the global alignment dynamic program is to create n by n matrix. N by m by n matrix. Here, if you see, this is 5 and this is 4, because this is already 5 and all. So you have to, if it is rows are m by n matrix, for that you should have m plus 1 n plus 1 matrix, that means 6 by 5. In order to solve, you should have 6 by 5 matrix. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. This is what you have to start. Then you have to fill the whole matrix, that is called scoring matrix. First step is initialization. Next step is scoring matrix. Third step is tracing back. Trace back. Fourth, alignment. Matrix filling. We will fill all the matrix with highest possible scores. We will try to fill all the possible scores. Actually, this one I am not taking. This one. And trace back and aligning will be done. We will try to fill all the matrix wherever match will come, and then we will do the trace back. This is what a dynamic programming. In the next lecture, Needleman-Wunsch algorithm and Smith-Waterman algorithm with example and solution will be shown.